Hello and welcome to Teaching Bio. Today we're going to look at some model full mark answers to common questions that regularly come up in the environment and ecology topics. So these questions are generally quite poorly answered by students, but there is a pattern to a lot of the questions, and a lot of these kind of questions come up in past papers. So it's a good idea to get a baseline understanding of what the Marx King criteria was then, and then use this and apply it in your exams. So first question. This is asking you to describe the method of mark release recapture for a certain number of species of organism in the lake. Now, generally in the exam, they'll give you some more context and they'll make you describe this process for a specific type of organism. So generally the marks are for firstly capturing a sample, marking it, and then releasing it, which is the first cardinal aspect of this method here. Then you need to think of a method of marking that is appropriate in that it does no harm, and it's always a good idea to write this down. Then you take a second sample and count how many marked organisms you can see within that second sample based on how many you marked in step 2, and then use this formula here, which is very important to memorize. It's important here that for this question, the examiners were rejecting references to transects. Another question is then looking at the consequences of leaching of fertilizer into ponds and lakes and thinking about the ecological changes as a result of that. So there's a nice stepwise answer that you can memorize. So nitrates and other ions effectively leach or run off into lakes. This then causes the blooming and growth of aquatic algae. Okay, it's important that you mentioned aquatic. As a result, once the algae start to grow, they effectively block out the surface of water in ponds and lakes. Therefore, other aquatic plants can't absorb the relevant photons of light, so they cannot photosynthesize, they can't produce glucose, and so they die. Bacteria or saprobionts or saprophytes will feed off these sort of dead organisms, dead aquatic plants, and other organisms that rely on the plants as a source of food, and then use up the oxygen within the water, okay? And that's through respiration. And eventually this will lower the concentration, causing dead water, which will obviously then indicate no sign of further life within that area. In terms of looking at the bacteria in more detail then, so we've got saprophytes or saprophytes, and they have varying mechanism as well with how the different bacteria break down the nitrogen within sort of dead plants. So we've got digestion and breakdown of protein, DNA, nitrogen substances, extracellular digestion, which is where there is sort of this exocytotic secretion of enzymes that are secretory, and they'll then break down the relevant parts. We've also got the formation of ammonia or ammonium, and then we've got elements of the ammonia cycle, which is a good idea to go over. Oxidation, remember, is the loss of electrons or hydrogen. This question now looks at the efficiency of energy transfer, and it's asking you to explain why and how the efficiency is actually different at different moments and steps of the transfer. And you're likely to get a sort of question where you get a flow chart and you need to then decide where there is the most efficient amount of transfer, and you might do some maths with this. So it's important to note that some light energy will actually fail to strike at a plan or whatever requires that light energy, or it's not the right wavelength, or it'll get reflected off. If you think about back to water, so you should ideally recall that green sort of wavelengths, they are able to penetrate the surface of water better, and so the seaweed and plants lower down in the sea will be red, so that they are appropriate wavelength. The efficiency of photosynthesis plants is low, or you can basically say that's about 2% efficient. You've got to always account for other causes of loss. Heat is always a really good one to put in. They love for you to know this concept here, that in respiration, energy is lost in the form of heat, okay? Lost in, very important to remember that. And again, the efficiency will then change as you go along the chain, and it's lower in the older animals and the higher up within that sort of pyramid of transfer. And again, carnivores will use more aspects of their food and then therefore gain more of those biological molecules from the food compared to the herbivores. We've then got intensive rearing of domestic livestock, and linking that to net productivity, which is always a bit sort of waffly, but it's simple concepts again. So intensive rearing of livestock How does that increase your net productivity ultimately? Well, we can kill them or slaughter them while still growing, so that's before they're mature, so while they're young. Therefore, we've got the most amount of energy being stored within the biomass or the tissue. And remember that energy is stored in the form of carbon in these carbon skeletons. It's important to note then that the domestic livestock that we have are able to feed on a certain diet, which often is injected with a sort of different materials like steroids and antibiotics, which could help. But then there's a counter argument for that as well. Again, controlled diet, controlled conditions. It sort of means that you sort of get more food being absorbed. We have less loss in feces. So sort of battery farming, cage farming, and so on restricts the movement, which then limits respiratory losses. So if movement is restricted, so how we actually get to this conclusion then, if we restrict the movement, then we do not necessarily need that much ATP, which remember comes from respiration, to actually cause the contraction of relevant muscles, to cause the movement. Therefore, we are conserving that energy that we would normally lose in the form of heat from respiration. So less energy is used, more energy is retained. And then that energy that's retained will help because as the animal is confined, there'll be less heat loss, so it won't have to accommodate by respiring more. And there's also the element of genetics. Then we've got succession. They love to ask about succession. They've started asking it in terms of sort of graphs now, but it's the same sort of concept. Make sure you contextualize your answers. And it's a simple process. So remember, pioneer species initially, they colonize the bare ground and they alter an abiotic factor. And it's a very good idea to name an abiotic factor because a lot of marketing are asking for that now. And therefore, that changes the conditions from being inhospitable to hospitable, allowing other species to then come along, compete, colonize, and survive. Then we pass through lots of different stages, and over time, we get this long, complex change in the biodiversity and an increase in the stability of the environment, eventually forming this climax community. Biological agents to control pests. Now, this has been taken off some mark schemes. However, they could still ask it in this sort of application concept. Advantages and disadvantages are quite straightforward. So generally, biological agents are specific to one type of pest. They only need to use it once or very little times. And it's hard sometimes for a lot of insects to develop resistance. However, they do like to ask about this. And linking it to genetics. Make sure you look into that. Disadvantages, it might not get rid of the pest. You can develop resistances or mutations, I guess. It takes time because you've got to allow the biological agent to pass through the population, and then those who aren't killed will then be able to reproduce or move on. The evolutionary mechanisms it might take to develop resistance might take time, so it can work for now. Then we've got a question on farming practices and increasing productivity, again, of agricultural crops. So again, agriculture, productivity, we're thinking all about energy and how we actually get that. So the importance of a named specific nutrient like nitrate or nitrogen for proteins, phosphate for ATP, and for that phosphate group in DNA. So we can have, again, the phosphate group within DNA. Got pesticides, which we talked about, weed killers, herbicides. Weeding will remove the competition. Again, they're competing for space, they're competing for nutrients, they're competing for water. So if we can remove the competition, yay for us. We've got selective breeding, genetic modification, greenhouses. Make sure you learn all the different limiting factors of photosynthesis. There's aspects of plowing, aerating the soil. That has come up a few times. And the purpose of ploughing effectively is this, and this is a very good idea to remember, and it's about nitrification and creating those conditions so that the nitrogen cycle, the ammonia cycle, and so on can actually occur. Crop rotation, again, passing around those sort of fertile soils and fertile nutrients and within all the different crops you have will increase the chance for the productivity of these crops. And again, we're back to limiting factors and then always about protecting it from predators. So we've got two more questions left. Next one is how the action of microorganisms actually produces the nitrate. So this is sort of more of the nitty-gritty of the nitrogen cycle. So important to remember that the protein, and more specifically the peptide bond within there, will be broken down into ammonia or ammonium compounds by saprobiotes. So for example, some type of fungi. The ammonium or the ammoniae will then convert into nitrate, which is known as a process called ammonification. Nitrite. So ammonium or ammonia becomes nitrite. Nitrite then becomes nitrate, and that's by nitrifying bacteria. Okay. And then nitrogen is converted to ammonia by nitrogen fixing bacteria. So it's a good idea to maybe make a table out of this. So nitrogen fixing bacteria, the reaction that catalyzes nitrogen to ammonia. Nitrifying bacteria will be nitrite to nitrate, which is NO2 minus to NO3 minus. And then the ammonia or the ammonium into nitrite, so NO2 minus, NH4 plus, NO2 minus, or NH3 will be ammonification. Chemical formula, be careful as long as you include the charges and you have the right numbers. And then last question, explain how pesticides can actually result in resistant strains. This is what we were thinking about before then. So generally what happens is there'll be variation and then there will be some kind of DNA mutation and you will confer that allele for resistance. It's important to note in the question how many copies of the alleles are actually going to be passed along, because they've started asking some questions now where they'll talk about multiple copies of an allele of a certain type of gene being passed on as a selective advantage. So it could be allele or alleles for resistance. And then that induces selection pressure and the better competitor will survive. And those, therefore, that have the resistant alleles will breed and, be better competitors. And then there's an increase in the reproductive success of these animals. We get an increase in the frequency of the offspring. And within that offspring, we'll have an increase in frequency of the relevant allele or alleles. Thank you very much for watching and good luck with your exams.